This is lecture 31. It's part 4 of 7 in anabolic cellular signaling. Let's start with this diagram. There should be a lot of familiar stuff here, a lot of familiar features in it. That long sideways yellow ladder, that's the phospholipid bilayer. Above it is the outside of the cell. So like the words muscle physiology, that would be the extracellular matrix. And then beneath the phospholipid bilayer, that's the inside of the cell. On the left, embedded in the membrane, you see an insulin receptor. When insulin binds to it, you get PI3K activation. And PI3K, it's a kinase that phosphorylates stuff. And what it phosphorylates is PIP2, right? Converting it to PIP3. But you also see PTEN here, PTEN. That's a phosphatase. So it removes phosphates from things. That's what phosphatases do. And this phosphatase removes a P from PIP3, converting it back to PIP2. So PTEN does the opposite of PI3K. But then once you have PIP3, the function of it is to recruit PDK, PDK1, and then PDK can phosphorylate PKB, AKT here, but AKT or PKB. Or it can indirectly activate mTOR complex 2. Downstream from that is PKB activation and complex 1 activation. But once PKB is on, it does a ton of things. Then also, it's worth noting, you don't need to know this, but just to acknowledge it, that PKB has 3 different isoforms encoded by 3 different genes. Um, but mostly what we're looking at is AKT1 or PKB alpha. That's the muscle hypertrophy one, AKT1. We also look at AKT2, right? That's insulin signaling, glucose transport. So like if you have a deficiency of AKT1, but AKT2 is fine, your glucose regulation will be okay, but you'll be tiny. And then you don't, but if you have a deficiency of AKT3, right, that leads to a smaller brain. Um, so these have different functions, these different isoforms of AKT, PKB, and just like there are different isoforms of myosin, right? But in the first block, I insisted that you know those different myosin isoforms. You don't need to know them for PKB. For the sake of this class, just think of PKB as a singular enzyme, a singular super anabolic enzyme. It leads to glucose uptake and glycogen synthesis, protein synthesis, tons of that. Cell survival and proliferation. It turns off catabolism, autophagy, and catabolism. So in protein turnover, anabolism is increased and catabolism is decreased. It doesn't just favor one side of the equation, both sides it addresses. So it's really anabolic and anti-catabolic. The only things here that you should be unfamiliar with that we haven't talked about and stuff that we're not going to talk about other than right this second, and you don't need to know about them, are those other phosphatases, PHLPP and PP2A. These are inhibitors of PKB. So PHLPP, this dephosphorylates PKB at one particular site, serine 473, it shows it. And then PP2A, that dephosphorylates PKB at a threonine site, 308. Other than that, and you don't need to know that stuff, everything here should be familiar. Okay, let's move on. We're going to cover both the endocrine system and nutrition today. We'll start with the endocrine system. And so think it like anabolic hormone supplementation. This is so effective. I mean, just look at Ronnie Coleman there on the right? That's a lot of hormones on board, exogenously administered hormones. And compared to the quote natural world champion there on the left, the word natural, the reason I say quote, it doesn't make any sense, right? Growth hormone, IGF, testosterone, these are all totally natural. But a lot of the supplements that both of these bodybuilders would be taking are made in a lab and don't exist in any form in nature, have never existed in nature. But the difference here is hormonal supplements. Clearly, hormonal supplements are anabolic, they work. And testosterone is the major androgen that we focus on that interacts with skeletal muscle. It's produced in the testes in males in response to the hypothalamic pituitary gonadal axis. So there's luteinizing hormone from the pituitary, which causes the testes to produce testosterone, right? Testosterone. And then women also produce some in the ovaries and adrenal glands. And the primary effect of testosterone is genomic. And the androgen receptor, what the testosterone binds to, this is in the cytoplasm. It's inside of the cell. And then once it's bound to androgens, it binds to androgen response elements, AREs, of genes. So again, testosterone has a primary effect that's a steroidal one, not cell surface receptors, but it binds to androgen receptors inside the cell. Then there's a conformational change in the receptor. It's transported to the nucleus, and it binds to the hormone response element and induces transcription. But testosterone also seems to enhance mTOR signaling. We see it enhancing mTOR signaling as a secondary effect. Uh, but there's like proposed mechanisms with G protein androgen receptors at the cell membrane, which activate phospholipase C, and that increases inositol triphosphate levels, which have receptors on the sarcoplasmic reticulum, which causes calcium release from the reticula, plural, which increases the intracellular calcium. And that seems to activate the MAPK cascade. You can inhibit this with G protein inhibitors. And so this seems to be well observed. But we'll talk more about calcium in just a minute. This is just another non-nucleic mechanism of testosterone. And then part of the mTOR activation with testosterone is through increased IGF-1 levels and then possibly enhancement of the IGF-1 signaling cascade itself. But we know IGF-1 triggers PI3K, and that turns on PKB. And then IGF also stimulates MAPK. IGF turns that on too, but it's mostly PI3K. And just a comparison of IGF-1 levels in the control group there on the left, the white bar, to the group that received nandrolone. It's anabolic steroid on the right, much more IGF in that group. And this study was just looking at hamsters. Um, but they found that the androgen administration, that it selectively caused hypertrophy in the higher threshold muscle fibers. So the type 1 and 2A fibers weren't as responsive, weren't as sensitive to the androgens as the 2X and 2B fibers. This is hamsters, right? So they have 2B. We don't have 2B fibers. Um, so summarizing the findings, part of the androgen-induced growth of skeletal muscle is through IGF-1 signaling, and then the high threshold fibers are more sensitive to it, to these hypertrophic signals. And we've known about the relationship between IGF and testosterone for a really long time, really-ish long time. So there's an article from 1993 here and another one from 2000 evaluating this. But the effect of testosterone on mTOR seems to be through both increased levels of IGF-1 and enhancement of the IGF-1 signaling pathway. Um, so again, two different mechanisms of growth with testosterone. There's the nucleic, right, the primary action of testosterone, and then there's the canonical cell signaling or mTOR activation. So two different ways that testosterone is working. And this diagram is showing that testosterone has its main effect by binding to its androgen receptor inside the cell. And then that hormone receptor complex enters the nucleus and binds to DNA. But secondary effects are the stimulation of PI3K, PKB, and MAPK. ERK on this diagram, that's MAPK. Same thing. And then something else that testosterone does is inhibit LKB1, liver kinase B1, LKB1. Don't think of it as a liver thing, it's ubiquitous. But this is that kinase activator of AMPK, which is arguably the most catabolic enzyme in the body. And we'll talk all about AMPK in the next lecture. But LKB1 is the primary activator of AMPK in skeletal muscle. Now, LKB1 is required for metabolism and function of the muscle. Um, Overexpression of it is catabolic. Too much LKB1 activity, catabolic. But knockout conditions don't really appear to be anabolic. Regulation does need to be maintained, but LKB1 is the activator of AMPK, and AMPK is extremely catabolic. It turns off protein synthesis and it turns on protein degradation. And androgens inhibit it. Androgens inhibit the expression of LKB1, which is another mechanism for promoting mTOR activity through testosterone. And estrogens do this too, but estrogen is up next. That's the next hormone we'll talk about. So I'll remind you of this effect in a minute. Now, both testosterone and estrogen also influence ion transport. This is another non-nucleic action. So if you've ever seen like World's Strongest Man competitions, nobody is lean. We talked about the bacon stuff in a previous lecture, high fat, high cholesterol, low fiber diets. These correspond to higher concentrations of steroid hormones, higher testosterone, and then higher levels of testosterone and estrogen will help facilitate better calcium mobilization, and then better calcium release is going to help with skeletal muscle function. So there is an immediate enhancement of performance that testosterone and estrogen both have. Getting into estrogen specifically, age-related losses of estrogen associate with age-related losses of muscle mass. So estrogen appears to preserve skeletal muscle through its primary steroidal genomic effects, through transcriptional roles. But just like androgens, there are secondary effects too. And one of these is that estrogens rapidly increase intracellular calcium. I promise to remind you of this. Here's my reminder. Intracellular calcium. Estrogens are helping facilitate. And then another role that estrogen has is the inhibition of tuberin. Now, it depends what cells we're in, but this is another potential anabolic role that estrogen plays, the inhibition of tuberin. This is another study examining the same phenomenon. That whole bottom line, that's REB, right? Ross homolog enriched in brain, right? Which is a GTPase. So when estrogen deactivates tuberin, it's activating REB, so mTOR can be turned on. So that's anabolic. But right on the other hand, like estrogen has tons of hands, and on one of its other hands, it promotes LKB1 and AMPK. So this estrogen hand is doing catabolic things. So estrogen is a bit more complicated than testosterone in terms of its cell signaling, in terms of its anabolic catabolic roles. But then moving on to insulin and IGF, you know how these things signal, right? It's mostly PI3K, PKB, mTOR, with some additional signaling through MAPK. And then growth hormone, right? How does growth hormone induce skeletal muscle hypertrophy? The HPL axis, right? Hypothalamic pituitary liver to release IGF, insulin-like growth factor, and you know how that works. So here's GH signaling, right? You see JAK-STAT. We've talked about that. You see MAPK on the far left, that ERK stuff. We've talked about that. And then you see PKB on the right. We've talked a lot about that. But really, how GH is promoting muscle hypertrophy is through IGF, insulin-like growth factor. And so, if you follow the signaling pathways, what you ultimately see getting phosphorylated is P70S6K and 4-EBP1, those downstream targets of mTOR. And you can completely knock out growth hormones' anabolic effects with either wortmenin or rapamycin. Now, remember, wortmenin blocks PI3K, and rapamycin obviously blocks mTOR, mammalian target of rapamycin. So that means this is a PI3K, PKB, mTOR growth pathway that growth hormone is going through. So, like, think if you are drive, like, let's say you live in Central California and you want to make the drive north to Portland or like south to Tijuana. You're probably going to take I-5, right? That's just the main road, but you can take other roads. Other highways exist, and back ways and whatever, but the bulk of traffic is going to be on I-5 for either destination. And that's how most of these hormones work. It's the I-5 of hypertrophic signaling is PI3K, PKB, mTOR. So if you take wortmenin, right, a PI3K inhibitor, and you take growth hormone, you're not going to grow despite the growth hormone. All right, moving on to thyroid hormones. These also stimulate PI3K, which is also abolished with the administration of either rapamycin or wortmenin. Okay, summary of the hormones, of the effects of these hormones, ignoring the genomic effects of testosterone and estrogen, just looking at the list of signaling pathways that these hormones go through to promote protein synthesis. And here's a summary of all three categories of mTOR activation that we've discussed so far, the immune system and sort of corresponding chemical activation, mechanical loads, and how that transmits hypertrophic messages, and then endocrine signaling, what we just finished talking about. So next up and last on our list is nutrition. So nutritional availability has a huge influence on whether mTOR can be turned on. Carbs, fats, proteins, these all work differently, and protein is the most important signaler, which makes sense, right? Given that mTOR is instructing the translation of protein, which is super expensive metabolically to do. And so if you don't give it the actual fuel, it's going to be tough for mTOR to do its job. If you don't give it the resources to do its translation, it's going to be tough for mTOR. So, to understand how, right, not why, but how nutrition affects mTOR, we have to consider the question, where does mTOR live? Right, the actual enzyme, the protein complex, where within the cell does mTOR live? And the answer depends on the nutritional state of that cell. If you deny a cell nutrition, if you starve it of glucose and amino acids, that mTOR complex is sentenced to walk the desert, right? It's diffused throughout the cytoplasm, wandering the cellular abyss. But then, if you present the cell with amino acids, RAG GTPases will shepherd the mTOR complex back home, right, to the mostly to the lysosomal surface, where it can return to its job of overseeing protein translation, overseeing growth. mTOR complex 1 can also sense cholesterol, right, which contributes to its translocation, helps it commute to the lysosomal work site. But the point is, whether it's carbs, it's proteins, it's cholesterol, the stuff you put in your mouth and like masticate a little bit and swallow, that stuff really affects mTOR activity, um, mTOR productivity specifically. So there are a couple of pathways amino acids go through, RAG proteins and then VPS34. Again, these RAG proteins localize mTOR to the lysosome, mostly the lysosome, where it can interact with Reb. And this is just mTOR showing up to the work site. But like one person, mTOR can't do the job alone, right? It's a team effort. Um, and so like RAG is mTOR's driver, right? It's the subway system that takes it to the job site or the taxi or whatever. And then REB is its secretary or manager or something like that. It's critical sidekick. So REB has to show up to the work site too. But let's focus on the stuff in this diagram. The purple stuff, that's mTOR, right? Red stuff, inhibition, green, activation. So you see regulator and RAG GTPases here. The TSC complex and VPS34. You see the lysosome. Lots of really important functions of the lysosome. But among them, it serves as a scaffolding, right? It's the work site. If the workers aren't at the actual physical job site, they're not going to be working. mTOR doesn't work from home. It has to show up, and the primary work site is the lysosome. It can localize to other places too, like mitochondria, endoplasmic reticulum, nucleus, right? But the lysosome is the primary work site. Um, the lysosome, though, it isn't just um, some like pointless arbitrary place where protein complexes have decided to gather, right, to do their business. Um, in the way that like a bunch of druids hanging out of Stonehenge or whatever. It serves a fun. It's more like farmers in their crop fields. Right? You can't harvest your crops from inside your house, or if you're out shopping or whatever. You like your feet need to be standing in the actual farmland, among the crops, and the same is true for mTOR and the lysosome. So here in this article, they're just talking about the RAG proteins. In the presence of amino acids, RAGs send mTOR to the lysosome. And in the presence of hormones, insulin, something like that, which tells cells carbs are coming. And in the presence of some growth factors and other chemicals that indicate a functional need to translate proteins, um, Reb goes to the lysosome too. And when the whole workforce is docked at the work site, at the lysosome, mTOR activity ensues. If only half the work site shows up, it doesn't really work, right? You need both inputs. You need nutrients and growth factors. So RAG localizes mTOR, complex one to the job site, which is where the PI3K pathway will be capable of activating it. When the growth factors are withdrawn and PI3K activity ceases, right, the tuberous sclerosis complex, tuberin, will shut down operations by inhibiting Reb. So swallow a bunch of amino acids, right? Especially like leucine, arginine, a couple of the important ones. Some amino acids are more important than others, and you'll increase the amount of GTP bound RAG, which binds to Raptor. The mTOR complex, and that facilitates interaction with Reb. Regulator, that's just RAG regulation. This is more detail than you need to know. Okay, lysosomes. These are amino acid storage sites, recycling regions. So, melt down your metal and make some new metal thing out of it. That's what the lysosomes are doing, except instead of metal, it's proteins. And this is non-selective protein degradation. And lysosomes, these only account for like maybe 2% or so of a cell's volume, but they're not much. Like mitochondria are 10%, 15%, something like, but lysosomes are only like 2%, but they're critical to metabolism and critical to survival. And one of those, critical duties is mTOR activity. All right, so technically there's RAG A, B, C, and D, and you need GTP loaded RAG A, but you can just think of this as one thing, RAG. That's okay. Add amino acids, and the mTOR complex can move to the surface of the lysosome. And the movement itself is kind of complicated. There's about 30 proteins involved, maybe more. Um, and they're putting it in proximity of Reb. Um, and then Reb directly binds to mTOR, which turns on its kinase activity. So you can induce mutations in rags that make it permanently on. And this is bad, right? Because the growth factors are just constantly turning on mTOR. In the absence of nutrients, you're still mTORing. And so regulation is lost. And so studies where this mutation is induced in mice, they don't live long, happy lives. Maybe they live happy lives. They don't live long lives, though, certainly. Okay, so what amino acids are we talking about? What amino acids are being sensed and contributing to RAG activity? I mean, there are 20 of these things. Do we need to eat them all, right? Or will a few representatives suffice? Turns out they're representatives, right? They're far more important, or at least much more recognized by the cell. And um, just like there's no one amino acid, right? That's going to do all of your mTOR activation completely. But leucine is the most important, and arginine would be its runner-up. Uh, just leucine and arginine by themselves, ignore the other 18 amino acids. They do a decent job of getting mTOR localized, but add in lysine along with leucine and arginine, and about 90% of mTOR activation you can get with just three of the 20. So that's not bad. The image to the right, um, T389, that's just a site on S6K1 or P70S6K that mTOR phosphorylates. So introducing different combinations of amino acids and seeing how much mTOR is phosphorylating P70S6K. If you want to add in a couple more amino acids, methionine and glutamine also effective. And so I would say those would be the five if you want to look, and we'll talk about that when we get into practical applications. But if you want to see the most important representatives, those are five good ones. So again, we don't need every amino acid on board. We just need a sample representing the whole. We do this in research, right? Inferential statistics. We take a sample of people and make inferences about the larger population. Right? Evolution didn't do a good job of anticipating chemistry labs and the ability to isolate individual amino acids. So if a cell sees methionine and leucine and arginine and that's it, there's sort of an assumption that all of them were eaten because nature doesn't really isolate. Chemistry labs do. But here, though, we're looking at a lysosome, and the lysosome isn't just an organelle that degrades proteins. Um, with an inviting surface. It's also a sensor for amino acids. So mTOR is given information about amino acid content in the lysosomal lumen, right inside of it. So you see SLC38A9, that's an arginine sensor inside of the lysosome, inside and outside. It's a transmembrane protein. But the lysosome isn't the only sensor, right, for amino acids. There are also cytosolic receptors, just receptors out in the cell. So RAG GTPases respond to both cytosolic and lysosomal amino acids. Um, and there's some stuff you don't need to know here. I'll say what they are anyway, right? So the VATPase, right? A vacuolar type ATPase. This moves proton, right? This controls pH. You don't need to know it. And then Kickstore, this recruits Gator. You can ignore that one too, but Gator one, don't ignore that. That's basically the tuberin of amino acid sensing. In the way that tuberin inhibits Reb, Gator 1 is inhibiting RAG proteins. But while PKB promotes GTP-bound Reb through a double negative, right, inhibits the thing that's inhibiting it, leucine and arginine are quadruple negatives, right? They inhibit the thing that inhibits the thing that inhibits the thing that inhibits RAG. So they turn it on, right? Let's start with leucine. Leucine binds to cestrin, right? Cestrin 2. Um, this is a cytosolic leucine sensor. Without cestrin 2, the cell seems to have no idea if you're eating leucine. So sestrin is what detects the presence and absence of leucine. With leucine deprivation, sestrin 2 will bind to gator 2. And then in the presence of leucine, sestrin 2 dissociates from gator 2. And then castor basically does the same thing that sestrin 2 does. It's just a different protein. But it too, right, inhibits gator 2. And arginine inhibits castor-1. Castor-1, this stands for CASTOR, cellular arginine sensor, CAS, for TOR, right? Cellular arginine sensor, CAS-TOR. So SLC 38A9 senses lysosomal arginine, and castor senses cytosolic arginine. Um, and again, castor will bind to gator 2, inhibiting it under arginine deprivation conditions. And if gator 2 is on, it's inhibiting gator 1, which is the direct inhibitor of RAGs. So if you want to grow, if you want to translate protein, you want gator 2 on. And cytosolic leucine and arginine both accomplish that. Another look at the same thing. Cestrin has a binding site to bind to gator 2, and leucine inhibits that binding. And then methionine, I mentioned that one also. This is also sensed very effectively through SAMTOR, SAMTOR. You see SAM, right, blocking SAMTOR, which turns on gator-1. SAM is S-adenosyl methionine. It's a compound that's derived from methionine. And SAM turns off an activator of the major inhibitor. So it helps mTOR turn on. And methionine seems like an obvious amino acid to sense, given that the start codon in protein translation codes for methionine. So it's the first amino acid in the manufacturing of every protein. So it's important to sense that one too. And methionine restriction is a pretty effective way to simulate protein starvation and prolong life in some animal models, and depending on conditions. Um, this is just a narrative summary of the basics of amino acid sensing. Eat your representative amino acids, and the RAG proteins bind to Raptor and relocalize mTOR so it can be activated. If you're short on amino acids, mTOR complex 1 and Reb dissociate. So insulin and IGF and all those mTOR promoters uh, in the presence of amino acid deprivation, they don't turn mTOR on. You need both of those things. So here's a nice diagram combining 4 upstream interactions with mTOR. EGF, that's epidermal growth factor. You don't need to know it, but just think of it as a general like growth factors that activate MAPK. So there's MAPK signaling, and you see RSK there, and the inhibition of tuberin. MAPK signaling, this also activates Raptor. And then you see PI3K signaling. This is the primary way of inhibiting tuberin. Then you see AMPK, which we'll talk all about in the next lecture. That's an activator of tuberin and an inhibitor of Raptor. And then you see RAGs, right, being activated by amino acids, and the RAGs are going to put mTOR in proximity to Reb to facilitate that interaction. So aminos to RAGs, RAGs to riches, if riches are proteins. Here's another diagram, lots of stuff you should recognize. Mostly, we're looking at the opposite effects of PKB and AMPK. Those two oppose each other. So on the left, where you see AMPK activating ULK1, ULK1, and stimulating mitophagy. We'll talk about that in the next lecture. Mitophagy is just autophagy of mitochondria, but we'll talk about that in the next lecture. But overall, it's a nice diagram. Other than P10, that's not quite what it does, right? It converts PIP3 into PIP2, but they're just saving space. Same reason PI3K appears to be activating PKB directly. Um, And it also looks like PKB is activating PRAS40. It's deactivating it. PRAS40 is a negative regulator of mTOR, but overall, this is a good diagram. And then it's worth acknowledging that LKB1 does lots of stuff, but the only thing we care about in this class is activation of AMPK. The far right arrow coming out of LKB1, that's the only thing you really need to know for the context of this class. This is another good one that combines both growth factor stimulation of PI3K, PKB, mTOR, and amino acid induced RAG activity and inhibition by AMPK and activation of PKB by mTOR complex 2. Um, if you ignore PRAK, right, that P38 regulated activated protein kinase, if you ignore that, you know everything else here. Now, in the last lecture, we talked about chemical and mechanical interactions. There are chemical and nutritional interactions as well. Things like reactive oxygen species and leucine. Reactive oxygen species mediate a lot of effects in the body, but just ignore these interactions and think of amino acid signaling as its own independent process, unaffected by some of these other phenomena. And here's our summary, right? Fats, carbs, and our summary of food, right? Fats, carbs, and proteins, and how they exert their effect. Now, we didn't talk about carbohydrates other than stimulation of insulin. And you know how insulin sends its signals through PI3K, but we'll talk more about carbohydrate in the next lecture and its effect on AMPK. All right, so let's review the big picture, right? The purpose of mTOR as we wrap up this lecture. So mTOR complex 1's purpose is to detect fasted and fed states. If you prevent mTOR from being inhibited when cells are denied nutrients, death is pretty swift. If you can't turn off anabolism, you can't survive a period of fasting. You just anabolize yourself to death. And if you think about most of our history, our ancestral evolutionary history, it's been marked by undereating and overactivity and not the other way around. As our ancestors, they spent a great deal of time in deprivation. And so, we have keen detection skills and quick responses for deprivation, for fasting, for food shortages. That's really what fitness, at least a large part of fitness, is matching our metabolism to our nutrient availability. I mean, that's a huge component of fitness. Um, Now, the categorical inputs of mTOR would be really need, nutrition, and assumption of nutrition. And we'll go through them one at a time. I'm not going to read the slides, but you can. And I hope it clarifies why mTOR is regulated in the way that it is. And this slide talks about need. If you're loading and damaging a cell, you're going to need to repair that damage. And so that's a major input for mTOR activity for it to be turned on. And the nutrition, eating the right amino acids will localize mTOR to a place within the cell where it can be activated. Assumption of nutrition, this is a little bit different. There are ways to make your body think calories are on the way. I mean, you can just put carbs in your mouth, swish it around a little bit, and then spit it out, and your metabolism will respond as though carbs are coming, even though they're already on the sidewalk. Because why wouldn't you swallow it? Right? So you can fool your cells into thinking nutrition is more abundant than it is. I mean, just eat particular amino acids, nothing else, and your body thinks you've eaten a whole protein. So little tricks are available for the assumption of nutrition. But we need both of pathways to activate it, right? We need to put mTOR in the right place with nutrients, and we also need to turn it on with growth factors. So mTOR is a two-key system. One mode of activation is insufficient to turn it on. It needs both modes to be turned on. So think of it as Gringotts, right, from Harry Potter, where the goblin and the wizard each need a key to open their designated vault. Or in real life, just think of it as quoting the Air Force instruction manual here, nuclear surety tamper control and detection programs, right? Section 1.1, right at the beginning, you need two people. One person isn't enough. It's too important for one person to activate. You need two. And the same is true with mTOR. It's a two-key system. And with that, here are questions. Here are the topics to pay attention to in this lecture. And I'll see you in the next one where we talk about AMPK and the negative regulation of mTOR.